Welcome to our channel Science Slide. Today our topic is about the DNA, that is the basic blueprint of life. As you know, all the data, or you can say the genetic data, is contained in the DNA. So today we will explore the molecular code that defines all living things, from the very basic bacteria to the complex human beings. Then the next slide we will talk about the discovery of DNA structure. The very basic step that was taken in the discovery was in 1950s, where Rosalind Franklin captured photos using the X-ray diffraction images, and then in 1952 Chargaff established base pairing rule, and you know that base pair rule is adenine always pairs with thymine, cytosine pairs with guanine. In 1953, then the Watson and the Crick unveiled the double helical model. This was the actual turning point in molecular biology, who proposed that DNA is actually double helical. So this was a breakthrough, revealed that how genetic information is stored and then transmitted, laying the foundation for the modern molecular biology. So here we will talk what is actually DNA. So DNA stands for deoxyribonucleic acid. It is the remarkable molecule that carries the complete genetic information or genetic instruction for building, operating, and maintaining every living organism on the earth. DNA actually serves as nature's instructional manual, containing the precise code needed to construct proteins, regulate cellular functions, and then pass the traits from one generation to the next. So it is primarily found in the nucleus of the cell, and it is also present in the mitochondria. It is also present in some viruses as well, and it contains billions of base pairs in the humans. So if we talk about the structure of DNA in detail, it actually contains deoxyribose sugar, a phosphate group, and nitrogenous bases. So if we talk about the DNA's building blocks, they are called as nucleotides. So what is deoxyribose sugar? Actually, it is a 5-carbon sugar molecule forming the structural backbone of each nucleotide. So phosphate group links nucleotides together, creating the sugar phosphate backbone of the DNA strands. Nitrogenous bases are, there are 4 types of nitrogenous bases: adenine, thymine, cytosine, and guanine. So there is a base pairing rule, as I said earlier, that adenine always pairs with thymine, A and T, while cytosine always pairs with guanine. These complementary bases pairs are held together by hydrogen bonds. So the double helix structure, if we talk about the DNA, it is double helical. There are anti-parallel strands. The two anti-parallel strands of the DNA run in opposite directions, creating structural stability. So the second one is sugar phosphate backbone. It forms the outer rails of the twisted ladder structure. And third one is the base pair rungs. So in the middle, like in ladder, you see the rungs are made up of, these are the rungs, these are made up of the base pairs. These base pairs are held together with one another by hydrogen bonds in between them. So, and if we talk about how DNA makes copies, or what are the steps in DNA replication. So what is DNA replication? It is actually a process that enables cells to divide and then transmit the genetic information from one generation to the next, or you can say making a copy of DNA is called as DNA replication. This precise mechanism ensures genetic continuity. So, first step, DNA strands are separated from each other. The DNA strands unwind at the replication fork as helicase breaks hydrogen bonds. So the next one is template formation. After separation of these forks, each separated strand serves as a template for building a new complementary strand. Then base additions. DNA polymerase is an enzyme that adds matching nucleotides following the base pairing rules. Then there were many models that were put forward in the DNA replication process. First model is termed as conservative model. It was proposed It proposed that the original DNA molecule remains completely intact while an entirely new molecule is synthesized separately. Then the second model is semi-conservative model. That is nowadays, we follow this rule. Each daughter DNA molecule contains one strand from the original parent DNA and one newly synthesized strand. So the third model, that was dispersive model, suggested that DNA strands would break into the fragments with each daughter molecule containing segments of both old and new DNA randomly disrupted. Why DNA is so important? We can say the importance of DNA is in hereditary and evolution. These are major things that are very important. So genetic inheritance. As you know that DNA serves as the molecular bridge between the generations, storing and transmitting genetic information from one parent to the offspring with remarkable fidelity. Each parent contributes half of their DNA to create a unique combination in their children, explaining both family resemblances and individual differences. So, it is very important to know that if we are talking about an offspring, each offspring gets half of the genetic material from parents, the male parent, and half of the genetic material from the female parent. Evolutionary engine. Mutations, random changes in DNA sequences that introduce genetic variation into populations, providing the raw material for natural selection. Over millions of years, beneficial mutations accumulate, driving the incredible diversity of life on Earth from single-celled organism to the complex multicellular beings. So, some interesting facts about the DNA. 9 million kilometers of DNA. If we talk about the length of DNA, actually the DNA is very, very long. The length of DNA in the average human body is enough to stretch from Earth to the Sun and back 30 times. Then 50% shared with banana. The 50% of our DNA is shared with banana. So humans share approximately half of their DNA with bananas, highlighting our common evolutionary ancestry. In 1869, first discovery was made by Friedrich Miescher, first isolated DNA from white blood cells, calling it as nuclein. So the important discovery is of Rosalind Franklin's photos that were taken by X-rays, that the critical breakthrough that revealed DNA's helical structure, yet her contributions were unrecognized during her lifetime. But today she is celebrated as a pioneer of the molecular biology. This was all from our lecture about the DNA. If you like today's lecture, please subscribe to the Science Slide, the YouTube channel, and share with your friends for more science videos.